Our next presenter is Joseph Opashevsky with Fujifilm Diosynth. And it's a change on the schedule that you see there. So the presenter is changing, but also the presentation is changing. And Joseph's presentation, Recent Advances for Rapid Development of High-Quality, Robust Mammalian Cell Culture Biopharmaceutical Manufacturing Processes. So with that, I'd like to introduce Joseph.
Okay, thank you very much. I will try to speak to the microphone. I cannot really talk too loud. It's already 2 days and been losing my voice. So please give me signs if you cannot hear me clearly. So essentially the topic today is rapid development of robust cell culture processes. I think the focus here is on both rapid and robust. A little bit about Fujifilm Diosynth for those who don't know us very well. So we global company. We have 3 sites, 2 sites in the U.S., one in North Carolina, one in Texas, and a site in northern England. We've been around for quite a while. We've worked on many different projects, many molecules, including 6 commercial products. Our range of manufacturing capabilities is from microbial production through cell culture through high containment manufacturing. And our cell culture range, we have cell culture capabilities at all 3 sites ranging from small scale to 2,000 liter, multiple 2,000 liter reactors. So the presentation is about acceleration of the process development for cell culture from the gene to the clinic. It's want to be clear, this is not about cutting corners, but rather making the timeline shorter while incorporating quality by design and making the process as robust as possible. So when we started offering our cell line development services, known as Apollo, approximately 3 years ago, the timeline from the gene to IND was around 18 to 24 months. Since then, we spent a lot of time putting innovation programs, putting new tools and new approaches to trying to shorten this timeline as much as we can. Among those approaches, we're starting from the actually rational design of the process through the tools that to shorten the cell line development and make it more robust. Then we also focus on tools for development of the upstream processes and more efficient development of the downstream processes. Of course, this cannot exist with robust analytics. So we spend a lot of time focusing on analytical development and putting in place quick and robust analytical methods for in-process testing. And finally, we focus on actual program management, risk management, and mitigation, because without this, the development and manufacturing cannot really happen reliably in the desired time. And our goal was to shorten the timeline to approximately 15 months or less. I will cover several of those tools today during the presentation. So it starts with the rational program design. We need to have a very clear target where we're heading with our development to meet our timelines and essentially meet the goals of development for our clients. So one of the tools that we put in place was a matrix for the phase-appropriate quality systems. This is very important not only for decision-making, for the scientists to make the correct decision, but essentially this is a tool facilitating conversations with our clients. For clients, some clients don't realize how important it is to know where we're heading, particularly for those who may anticipate accelerated approval or breakthrough designation for the molecules. The rational design of programs started where we were essentially developing the Apollo cell line system. When we were focusing on goals for the cell line development, the high titer was not the actual objective. The objective was to get a cell line that is suitable for manufacturing and can be taken all the way to commercialization. So hence the cell line for life. Part of this development was, of course, developing a platform media for the cell line that can accelerate and other elements of the cell line development process. We also look at the entire process from the Apollo to manufacturing. And we also realized that one of the bottlenecks in development is essentially cell banking. Very often it's disconnected cell banking from the development. It's essentially delays development. We put our own cell banking facilities that essentially allow us to feed the cell banks when it's needed for the development. All right, so that was about the rational design. Now about the actual tools for acceleration of the timeline. So the first set of tools were around the cell line development. So we were looking how to shorten the timeline. And we implemented two major innovations that allow us to achieve the goal. So the first was to go away from the static plate and introduce the fed bar steered plate. And the second innovation was replacing the shake flask with amber 15. The steel plates really performed very well. We did comparison with the shake flask, and we essentially were able to achieve the results similar to the shake flask by using multi-well format in a very small amount of culture. This really saved time on growing the cells and basically on the ability to screen a large number of samples. We use amber 15 mostly for the final clone selection. And once again, we're focusing here not only on looking at the cell growth or product titer, but we're also looking at the quality attributes. We implemented a robust in-process analytics that I'll be talking about in a moment to essentially facilitate this. So we make decisions not only based on titer, but based on the actual desired product profile. Another area of development where we actually invested quite a bit through innovation was developing tools for upstream process development. And during the innovation program, we screened over 60 media and almost 20 feed strategy and selected those that we believe that are more promising. So the current version of the toolbox, it's based on the 4 basal media and 4 feeds. So the toolbox essentially works this way, that basically we adapt cell lines to the media, then perform the high throughput screening in the amber 15, use the high throughput analytics, TICAN, which I show in a moment, to get quality attributes, and then we select the clones and we select the media desired set of media and feeds and confirm those in the bioreactors. It's rapid and efficient because we focus on really limited number of basal media and feeds. This is just an example of application of our toolbox. So the problem statement was we had the process developed in 2007, then required media refreshment. So we applied the toolbox, and this slide shows that we essentially significantly boosted cell line productivity while also increase viability of the cell line at the end of the production. And we did this without impacting critical quality attributes. We screened a number of those. This is just one example looking at glycans. So the output of this was actually that we doubled the original titer production of the protein. From something that in 2007 we believed we had really, really good process that was giving us 2.5 to 3 grams per liter, we actually managed to boost this to over 5 grams per liter just using this limited screening with the toolbox. All right, this was the upstream. So another set of tools we implemented around the downstream development. So once again, looking at high throughput, looking at rapid tools. So we implemented automated high throughput screening for media, for buffer column elution or buffer conditions, looking at the protein stability and protein attributes. In this example here, we actually look at the hydrophobic interaction chromatography. We look at the conditions for protein, for elution from the column and on protein stability. We look at the ammonium sulfate concentration, pH. We look at protein concentration. And we selected areas that were safe to operate or desired area to operate on the hydrophobic interaction chromatography. Then we use those conditions and apply to the robo columns where we actually screen different sets of resin. In this case, this example shows the 5 types of resin screened over 3 different pH conditions. Once again, using automated robotic system. The advantage is obvious. It's not only reducing the volume of the material needed to microliters or milliliters, but dramatically reducing the timeline. Of course, none of those will exist without analyticals, with correct analytics. So we spend a lot of time looking at analytical development. And actually, tomorrow, Greg Adams, head of analytical group at Fujifilm, Diosynth, will be presenting his view and our approach to analyzing the proteins. So throughout the development, we essentially look comprehensively at different protein attributes. We look at the charge heterogeneity, we're looking at glycosylation heterogeneity, we're looking at aggregation, oxidation, fragmentation, we look at post-translation modification using a wide number of tools. Of course, the challenge is quite a bit, because if we're using the amber, let's say we're using the amber with 48 reactors, and we actually look at this protein quality attributes over 5 days, which we typically do 3 to 5 days at the end of the culture, we're generating hundreds of samples over the couple of days that we want to analyze with a range of tools. Fujifilm has historically been very strong in analytics. We have pretty much comprehensive analytical methods in-house, and we continuously update our analytical base, trying to really get the newest technology, and whenever we can, we really bring those in the high throughput format that facilitate in-process control. Of course, with this number of samples and high throughput analytics, we also need high throughput sample preparation. This was actually one of the first projects that we did in this area, implemented a TICAN sample preparation. So we essentially can prepare hundreds of samples within less than 2 hours and submit those to the analytical testing. And then using rapid analytics, that basically the target for those is, rapid means less than 10 minutes per sample, we can essentially turn around hundreds of samples within 1 day. So the target that we routinely achieve is pretty much 8 hours turnaround. So essentially the upstream scientists and downstream scientists, they getting the real data the same day. So they can actually rationally design the next step in the process. So this is real QBD. You essentially drive the process through data and analytics. Those are just examples of some methods that we use. For example, as I said, the target is all analysis in less than 10 minutes. So size exclusion, UPLC, typically 4 or 5 minutes. The same, the mass spec, it's a few minutes, and some of those were even faster. Okay, so this is real QBD. Now, if you think about this, I started the talk talking about the shortening the timeline. Now, what we've really done was we shortened analysis, we shortened some period or some experiments, but we're adding such complexity to this process that we're generating much, much more data than we used to. So what we also needed is essentially we needed effective program management and effective tracking of the process and identifying and mitigating the risks that are associated with this. So one of the things that we implemented in program management is efficient and very effective formalized setting a milestone, locking milestone on a regular basis, essentially weekly, and tracking those milestones. Another thing that we implemented was essentially a number of tools for identifying and mitigate risk. We try to use the platform process as much as we can. And of course, platform process big advantages. It reduces the risk and allows us to focus on things that basically are different. We know that each molecule will be different. Sometimes each cell line will be slightly different. So what we're trying to focus our risk management on new, unique, and difficult, what we call NUDS. This allows us really to have a focused risk assessment. We have focused risk assessment in implementation of mitigation strategies. We regularly perform potential problem analysis before any manufacturing campaign. And finally, all this is structured into the formalized stage gating process. So the stage gating process allows us not only monitoring timeline, but making sure that the entire process goes in the right direction, and basically the risk to the manufacturing at the end is minimized. So the stage gating, we have stage gate at every single important milestone in the program. And basically, we actually have a voting process where the site leadership team essentially votes every time on readiness of the process to go to the next stage and readiness for the manufacturing. All right, just using all those different tools, we essentially achieve our goal of reducing the timeline to 15 months, while we also managed to implement really quality by design principle and essentially generate very robust processes without any cutting corners. So in the case of the cell line development, we essentially reduced the cell line development time from 9 months to 6 months just by implementing those tools, while we know now we have much more information about the cell line itself. In the case, those other tools that I discussed allow us to essentially have the process development time, while once again generating a lot of information about molecule behavior and rationale for the process development. So essentially that was the example that what we do for implementation of quality by design while reducing the timeline. And I think that was all what I had for today. Please visit us at our booth, which is right there.